Hello everyone. Today I would like to present a study about comparison of extracellular voltage distribution in cochlear implant system with and without carrier, a computational study based on microcity images of a human cochlea. Before I go through the details, I would like to explain some generals about hearing process. As you can see in the figure 1, the ear can be divided into three parts: the outer ear, middle ear, and the inner ear. Sounds travels down the ear canal, hitting the eardrum, which causes it to vibrate. These vibrations cause movement of the fluid in the inner ear that impact on the hair cells, which sends the electrical signals from the inner ear up to the auditory nerve. When the auditory nerve is damaged, the cochlear implant can be helpful. Cochlear implant is a surgical device that is inserted into a scala tympani. But we are in this study, we use human microcity images to segment the cochlea and 30 auditory nerve bundles. Then a 3D finite element model was built based on the segmented data. In addition to that, two cochlear implant systems were simulated, one lateral close to the lateral wall of the scala tympani, and one perimodular array which is close to the medial wall of scala tympani. But here in figure 2 showed only lateral system. Figure 2a shows the whole cochlear implant with carrier, and figure 2b shows only the electrode. Here, we compared potential distribution of three electrodes from each cochlear implant systems in the presence and absence of carrier. Figure 3a shows lateral system and figure 3b displays perimodular. The left panel belongs to the closest nerve to the electrode in the basal. In the middle panel belongs to the closest nerve to the electrode in the middle ten, and the right panel belongs to the closest nerve to the electrode in the apical nerve. As you can see in figure 3, when the carrier is added, potential value decreases by a shift. The reason for this is that the surface of electrode in the carrier system is decreased by 75%. But for validation of the model, we compared the intercochlear potentials with clinical data. Figure 4 shows that both cases with and without carrier are close to the reported clinical data. And in conclusion, in the computational models, if you want to consider all details, it would be very time consuming. So you need some simplification in the model. These simplifications should not affect the reliability of the main result. Here in this study, when we simplified our model to only electrodes, you can see the extracellular voltage only shifted a bit. And this shift in final equations mostly won't affect the site of action potential in our model of neurons. Thank you for your attention.